All right, guys, welcome to class. The beauty of technology is that you get to have a class while I'm not even there. So let me put this in full screen for a second. If you'll note, there's your homework. So section 513 to 516, pages 83 to 85. If you're in block 2, at the end of this, you'll have time to start working on this. Block 6, you guys probably see me for a second block today, or you'll be back in class, so you will have time to work on this and then move on to your USG. Either way, we're going to have a mini quiz on enzymes. Probably early next week. Thanksgiving is Thursday, so you guys have a half day Wednesday, so we'll have a quick quiz on this information before you go so you don't forget it all after your turkey. Alright, so moving into enzymes. We've already talked about this a little bit, so hopefully some of this stuff sounds familiar. Alright, what are they? We've already talked about this with macromolecules. So most enzymes are protein. Not all, there are always a few exceptions. You don't need to worry about them. If you want to look into some research, you can take a look at ribozymes. So there are a few things that can catalyze reactions that are not enzymes, but it's kind of new in the research. So for our purposes, all enzymes are proteins. They catalyze or start reactions in your body. So anything that you can think of, digestion, any of your metabolism, breaking things down or building things up would happen too slowly without it, so we need an enzyme. Luckily, they're not permanently changed though, so once you use an enzyme, you can still reuse that enzyme after it's done whatever it's doing, either building or breaking down. Alright, moving right along. So enzymes are very specific for what they catalyze. So if you notice in that cartoon picture, it's got a very lock and key mechanism. So whatever it is that it is binding is going to be very specifically shaped to match the enzyme itself. We know this, structure and function. So they're reusable. And remember that all enzymes end in ASE. Of course, again, there's exceptions. So things like pepsin is in your stomach. That doesn't end in ASE. In terms of your test, if it doesn't end in ASE, I will tell you that it's an enzyme. Otherwise, you should recognize it by its suffix. So knowing all this, what do you think lactase catalyzes? If you were thinking lactose, you are correct. So lactose ends in OSE, meaning it's a sugar. So let's take a look at this diagram on lactose intolerance. No ice cream, pretty sad. So in normal digestion, on the left there, normally you would take in lactose drinking milk or yogurt or any dairy product. Your body would hydrolyze that, right, hydrolysis reaction, and break it down into its two monosaccharide monomers, glucose and galactose. Those would get absorbed into your bloodstream, sent off to your mitochondria. We make ATP energy. For people who are lactose intolerant, they don't have the enzyme lactase. So what happens is the lactose stays in your body, and bacteria that live inside your gut will then eat that lactose. When they do that, because you're looking at anaerobic bacteria, they don't need any oxygen, they don't breathe the same way we do. So when they, quote unquote, breathe, they release acids and gases and all sorts of unfun stuff that causes you to either have really bad stomach aches or, in some extreme cases, enough acid that'll make you throw it back up. Pleasant, huh? Alright, moving on. So how do they work? They work by weakening bonds, so they kind of put stress on the bonds that already exist so that when you try to build something or break something, they're already essentially halfway done. So enzymes weaken the bonds and lowers activation energy. Excuse me. Activation energy is the amount of energy it takes for any reaction to run. So take a look at that graph there. On our x-axis is the progress of the reaction. So we want to start with those two reactants, AB and CD. We want to end up with AC and BD. But in the meantime, you can't just poof, snap your fingers and make it happen. There's a little bit of energy required. The amount that's required is on the graph there as EA, so that's your activation energy. Think of it like a roller coaster. If you started a roller coaster without going up first, you're not going to go very far, and even if you do, you're not going to make it to the end. So enzymes lower how much activation energy it needs. It's more energetically efficient for your body, and you get your products much more efficiently. All right, so you should be following along on your PowerPoint slides there, and it should tell you to graph what it would look like, a reaction would look like with and without an enzyme. So I'm going to zoom ahead here, assuming that you've already tried. Here is with, right? We've got that hump there for activation energy, your reactants, and then your products at the bottom. With an enzyme, we've lowered the amount of energy it takes. Very simple. All right, a few vocab terms just so that we can all be on the same page with this. The enzyme substrate complex. What is it? So a substrate itself is essentially another word for reactant. It's whatever thing that the enzyme is working on. So the substance or reactant that an enzyme acts on is called the substrate. There it is. Don't make fun of my enzyme there, I tried. So, when our substrate joins the enzyme, when the reaction needs to take place, we have an enzyme substrate complex. All right, here we are. We're joining the enzyme. The place in which that it actually joins is called the active site. So a restricted region. In this case, restricted doesn't mean a bad thing. It just means a very specific spot on the molecule for which the substrate is going to bind. Again, highly, highly specific. So it'll fit lock and key. If the substrate has a square key, the enzyme is going to have a square keyhole. It doesn't fit perfectly, it's almost perfect. So an enzyme can kind of fit around. Remember, enzymes are globular proteins, so they literally look like blobs. When they bind, it does sort of cuddle the substrate so that it fits in there nicely. And there we go, there's the active site. All right, things that affect enzyme activity. 3 major categories: environmental conditions, cofactors and coenzymes, and enzyme inhibitors. What does all that mean? Let's start with environmental. Things that come from the outside that would somehow impact it. We've already talked about one when we talked about proteins denaturing. When proteins denature, they often do so because of extreme temperatures. That tends to be the biggest issue that an enzyme runs into, aka fever. So high temps will generally denature, fancy word for unfold, that enzyme, and again, structure determines function, so it no longer will work. Second condition, pH. So generally, hopefully you guys had this in middle school, but things that are under 7 are very acidic, things that are over 7 through 14 are basic. Most enzymes like to hover around neutral, about 7, or water. Again, there's always a few exceptions. So enzymes in your stomach has hydrochloric acid in it, which is about a pH of 1, maybe 2. So those enzymes in your stomach are, of course, going to like a much more acidic environment than things that you would find throughout the watery cytoplasm of your cells. And your last one, ionic concentrations, mostly salt. In your body, you're looking at things like electrolytes. Remember that your brain runs off of electricity, so it needs positive and negative ions floating around in there in order to get all the messages where they need to go. So salt interacting either too much or too little with those enzymes are going to play a role as well. All right, condition number 2, cofactors and coenzymes. So sometimes the enzyme by itself is not enough. It needs a little help. So inorganic substances, aka things without carbon, like zinc, iron, any of your vitamins, minerals, sometimes those are required in order for your enzymes to properly bind whatever they need. Hemoglobin is going to be a really big example. So the inside your red blood cells, what carries the oxygen. In order for oxygen to actually bind to the molecule, iron has to be present. So the attractiveness between iron and oxygen has to be there, otherwise that globular hemoglobin protein won't carry anything. All right, last topic. So enzyme inhibitors. Inhibitor means to stop something. So we've got a couple things in our bodies that prevent enzymes from doing their job. This isn't always a bad thing. So sometimes you need to regulate how much an enzyme is working. So sometimes it's a good thing. But our first one are competitive inhibitors. So these guys are chemicals that somehow mimic the enzyme's normal substrate and shape, and they compete directly with it for its active site. So if we take a look at our animation here, there it is. There's our competitive inhibitor binding to the active site of the enzyme. Except now our substrate can't. So there are enzymes with multiple binding sites, but generally we're not talking about one substrate, one competitive inhibitor. There's a lot of them. So our competitive inhibitor is bound. Once it unbinds, the substrate can go in, but of course it takes some time. And again, not always bad. If you've ever heard of ALD, adrenoleukodystrophy, or watched the movie Lorenzo's Oil with Susan Sarandon, that was a true story. ALD is a condition in which there are enzymes in your body that attack the fatty acid tissue around your neurons. So your brain cells are coated with these nice soft pillows of fat to help the messages go through. If your body starts breaking it down, then the messages don't get there. So you start having outbursts, eventually then leading to blindness, loss of hearing, loss of senses, paralysis, and ultimately death. So what we did, or what researchers have done, is they figured out what the competitive inhibitor to that enzyme was. So we found out that if we pump these patients' diets full of these very particular fatty acids, we can keep the enzyme busy, so to speak, breaking those down so that they don't actually break down the fat tissue on your cells. Alright, if we've got competitive inhibitors, we most likely have non-competitive inhibitors. Competitive, they directly go head-to-head with the substrate for the active site. Non-competitive are a little bit more sneaky. These guys do not enter the active site. They bind to another part of the enzyme. The other part of that enzyme is called an allosteric site. So it binds to some other location. And when it binds, remember how I said that the enzyme kind of cuddles the substrate? That ultimately changes the shape of the whole enzyme. So when the non-competitive inhibitor binds, it changes the shape of the active site so that when the normal substrate comes along, it doesn't have a place to go. Again, not always a negative thing. Generally, when you have feedback loops, so if you have too much of one thing, a non-competitive inhibitor might bind so that your body does not continue to do whatever it is doing too much of. Remember, right now we're only talking about enzymes and substrates sort of in general. You guys will be doing a lab where you'll have a very specific enzyme and very specific substrates. So this is just sort of the summary overview of how these guys work. If you guys would like to go to this video clip here, if you don't want to type it in, you can search Google McGraw-Hill How Enzymes Work, or I've posted a link on the wiki space at the bottom of the biochem page so that you can go and kind of watch this in motion, the substrates going into the active site, binding, what happens, and releasing. If you have any questions, please make sure you write them down as you go on your note guide there so I can adjust them when I come back. Feel free to play this as many times as you want, go back, go forward, add in your notes, and then anything you guys still need clarification on, I'll make sure I address that when I see you guys tomorrow. So again, make sure that you have your homework assignment written down, 83 to 85, double-sided sheet of notes with summary as per usual. You guys can start that now, and I will see you tomorrow. Have a great day, everyone.